And so in the previous lecture, we went through the process of meiosis, and really through the process of meiosis, that's how it achieves this first goal, right? So the two divisions in both meiosis 1 and meiosis 2, where the DNA is not replicated in between, serves to help reduce the chromosome number from diploid to haploid, or cut it in half, right? Which is one of the main goals of meiosis. The other main goal is to create genetic diversity, right? And there are two main ways that meiosis does this. And we talked about the first one in the previous lecture as well. And it's through crossing over. And so when you have two homologous pairs, or a homologous pair, you see here a blue and a red chromosome. And then you allow crossing over to happen between the blue and red chromosomes, you can actually generate kind of a mixed chromosome, right? That's a little bit red and a little bit blue. And when those ultimately become gametes, that mixing of either maternal and paternal DNA during crossing over helps to generate some diversity in the gametes, right? And the other way that meiosis creates genetic diversity is through this idea of independent assortment, which you can see here on the right. And this is a concept that I always struggled understanding, but really what it means is just that these chromosomes will assort independently of each other. And whether you end up with a red chromosome on the right or a red chromosome on the left group of cells is just totally random and due to chance. There's no way to predict it, and there's really no difference. But the chromosomes can line up so that all the blues are on the left and all the reds are on the right, or they can line up like this, where there's a blue bigger chromosome on the left and a red one on the right, and a small red one on the left, blue one on the right. And that doesn't matter at all. And this idea of independent assortment, you can see leads to different gametes in the end, right? So if you look at this first scenario here, the gametes generated on the right have completely red chromosomes, or say all maternal copies of the genes. And the chromosomes on the left have all blue chromosomes, right? All these gametes have just blue chromosomes, or have just paternal DNA. But in this scenario on the bottom, this gamete actually has one blue chromosome and one red. And these gametes over here have one red chromosome and then the small blue. And these differences in how the chromosomes basically line up and who's on the left and who's on the right generates genetic diversity as well. Because if you remember, really what we're talking about is a copy of mom's DNA, or maternal copy, and a copy of dad's. And so sometimes you end up with gametes that contain all dad's DNA, or all mom's DNA. And you can also end up with gametes that have a mixture of mom and dad's together. And that is due to independent assortment, or basically just how they line up during metaphase. And so ultimately, through meiosis, multiple goals are achieved, right? One diploid cell is divided into 4 haploid germ cells or gametes. And then genetic variation is created during meiosis by crossing over in prophase 1 and by the random assortment of chromosomes in metaphase 1 and metaphase 2. And to compare meiosis to mitosis, remember from mitosis that we take a diploid cell, which has 2 copies of each chromosome, we duplicate the DNA in S phase, and then we divide once to create 2 identical daughter cells, both of which are also diploid. There's only one cell division, and one diploid cell makes 2 identical diploid daughters. Whereas in meiosis, one diploid cell duplicates its DNA during S phase and then goes through 2 divisions, right? One division here and then another one here. Meiosis 1 and meiosis 2. To divide one cell into 2, and then ultimately those 2 into 2 again to make 4 non-identical haploid daughter cells, right? And so meiosis has 2 divisions, makes 4 non-identical cells, and mitosis has 1 division and makes 2 identical cells. And so I'd like to have you guys take a second and answer a couple questions just to make sure that we are all on the same page, right? And so take a second and answer which phase of meiosis crossing over occurs in. Pause the video, take a second. All right, and the answer is A, prophase 1. So crossing over happens almost immediately in the first phase of meiosis 1, which is prophase 1. It does not happen again in prophase 2, which is important to remember. Okay. During which phase of meiosis do the sister chromatids separate from each other? Take a second, pause the video, think about it, decide. So you're likely trying to decide between C and D because the separation of DNA happens during anaphase. And so there's anaphase 1 and anaphase 2. And that's where the sister chromatids comes in to be important to answer the question. Because the sister chromatids actually separate during anaphase 2. Homologous pairs, or the two X's, separate from each other in anaphase 1. The two halves of one X, or the sister chromatids, separate from each other during anaphase 2 in that chromatid disjunction. And so we've talked a lot about meiosis and the process and the goals of meiosis, right? And so it's important that meiosis is done correctly to achieve these goals, right? You want to make sure you have the number of chromosomes in your gametes and you've generated some genetic diversity. But when it goes wrong, what can result is a thing called aneuploidy, right? And so euploid is a normal number of chromosomes. Aneuploid is a basically not normal number of chromosomes, or a numerical change in part of the genome, where you have one extra or one less chromosome than you should. And if you're hypoploid, that particular chromosome is underrepresented, or you don't have as many copies as you should. And if you're hyperploid, you have too many copies, or one extra of each chromosome. And what's important to note is that aneuploidies never really apply to all the chromosomes. If you had extra copy of every chromosome, it's likely that the zygote formed in that situation would not make it, would not be viable, right? So this is talking specifically about one particular chromosome that either has too many copies or too few copies. And I can show you some of the disorders that result from aneuploidy, or having an overrepresentation or underrepresentation of specific chromosomes, right? And so one example that you've likely heard of before is Down syndrome. Down syndrome is an aneuploidy. Specifically, it's what's called a trisomy. So tri is 3. There are 3 copies of a chromosome, in particular chromosome 21, and this occurs in humans, right? And so 3 copies is one too many. It's an extra copy. And this results from problems separating the chromosomes correctly during meiosis. And so it happens that they either don't line up correctly or they're not separated correctly during anaphase, and that can lead to an extra copy of chromosome 21 in some cells, right? And then if one of those is the gamete that is used to make a zygote, it can end up causing Down syndrome. And the frequency of problems in meiosis, like the ones that cause Down syndrome, actually increase with maternal age. So you can see here the risk of developing Down syndrome is also associated, is kind of correlated with an increase in the mother's age as well. And that's probably because meiosis becomes more difficult as we age, and so these problems with meiosis become more prevalent and can lead to an increased chance of having a child with Down syndrome. And some of the main hallmarks of Down syndrome are intellectual disabilities. There are some characteristic facial appearances, some changes in muscle tone, and it's kind of a spectrum disease, so there are usually cognitive delays associated with it. They can be varying between individuals. And there's also an increased risk of developing a lot of other kind of severe medical conditions, like hearing and vision problems, thyroid issues, as well as higher risk of developing Alzheimer's disease. And so you can actually see here the chromosomes. So there's 2 copies of every chromosome in humans, right? They exist in pairs because we are diploid organisms. And you can see here at chromosome 21, there's actually 3 copies. So this is a person with Down syndrome's kind of chromosome karyotype or map. And you can see how that might result from problems in meiosis here, right? And so if there's an event where the homologous pairs don't separate correctly, you can generate some gametes that have too many copies of particular chromosomes. And that can happen in the first part of meiosis, and it can also happen later in the second part. So in this scenario, the sister chromatids don't separate correctly, and you can end up with a gamete that has too many copies of the chromosomes as well. So another example of a trisomy, or a syndrome where there are too many copies of one chromosome, is a syndrome called Kleinfelders, where there are 3 copies of the sex chromosomes, particularly 2 X chromosomes and then a Y chromosome. Right, so normally you either have 2 X's or an X and a Y, and that determines your biological sex. So 2 X's being biologically female, X and a Y being biologically male. And so individuals with Kleinfelders are phenotypically male because they have the Y chromosome. And then they develop some female secondary sex characteristics, such as the development of breasts, right? And so people with Kleinfelders are sterile, and Kleinfelders is usually coupled with an intellectual disability as well. Some people with Kleinfelders can have issues with motor skills and other types of locomotion. There is a large association of this intellectual disability with also kind of reading issues, delays in speech and language development. Yeah. But it's also important to remember that it's not just having too many copies of a chromosome that can cause a disorder. Too few copies can cause some disorders as well. And one example of that is here with Turner syndrome. And this is where you only have 1 sex chromosome. In this case, you have 1 X chromosome, and you don't either have the additional X to make you biologically female or the Y to make you biologically male. And so because you only have 1 X chromosome, phenotypically and biologically you are female, you lack a Y chromosome, but you also don't end up developing ovaries, right? And so individuals with Turners are sterile as well. They have this characteristic webbed neck phenotype, which you can see here. There's also sort of this low hairline. Individuals with Turners can develop swelling of their hands and feet. They can develop issues with their skeletons, kidney problems. A lot of people with Turners are born with heart defects that can cause them health issues or cause health issues later on in their lives and actually be quite life-threatening. But ultimately, in terms of developmental defects, they seem to be less characteristic in Turner syndrome than they are in Kleinfelders or in Down syndrome. Right? And so it's important that meiosis is done correctly, otherwise you can develop these different syndromes or disorders when meiosis is kind of done incorrectly.